Okay, lecture 9, we'll have a discussion on DNA sequencing analysis. So why is that DNA sequencing analysis important? So the reason is the ultimate nucleotide sequence that's found within your DNA segment actually determines the genetic information that is carried by the DNA segment. Okay, so the order of the nucleotide, GATC4 nucleotide, how they exist in what sequence order, that will determine what RNA molecule will be made through the transcription process, and then based on that RNA molecule sequence, it will determine what protein molecule will be made. You would remember that codon table from your previous biology class. Yes, triple nucleotide codon will call for a specific amino acid. So understanding the sequence of nucleotide found from your DNA is very important because of that. Also, you need to understand the start and stop part of your gene. There's a gene coding sequence within your genome, right? So there are so many genes, maybe 30 or 40,000 genes found in your genome. Then you will have to understand which part of the DNA is encoding a certain gene. Okay, so once you understood that, gene coding region, you could also manipulate that gene coding sequence if you want to. Maybe you can clone that gene and get it out of the human genome, for example, like insulin gene, and you can cut and paste with a foreign DNA, maybe a DNA from bacterial cell like plasmid DNA, to construct recombinant DNA, and then you can move that recombinant construct into a new host cell like E. coli, so you can make more of the insulin protein using a bacterial cell strain. So you could do those kind of manipulation if you understand the DNA sequence, of course. Okay, and then construction of a restriction map of a gene of interest. What does that mean? If you want to cut and paste your DNA sequence, you need to know what enzymes will cut your DNA sequence out, right? So there are multiple enzymes that can recognize and cut very specific DNA sequences. So in order for you to be able to do that, you need to know the sequence of your DNA. Otherwise, you cannot designate the segment where your DNA will be cut. And a restriction map allows subcloning of a gene of interest into various vectors. Like I said, it's a gene cloning process. Once you cut your DNA, and that cut DNA can be recombined, in other words, repasted with another DNA fragment that has the matching ends. Okay, that's a ligation process. You could do that. And identification of coding regions and 5' to 3' and non-coding regulatory regions of a gene and is only possible after understanding DNA sequence. What does that mean? If you're looking at the eukaryotic gene coding sequences, okay, there are so-called exons and introns. Exons are coding regions, okay, but introns are non-coding regions. Okay, if this gene from here to here is transcribed into an RNA, and that RNA will be processed. Processed means, those non-coding regions, intron copies will be cut off, and only the exons will be put together. Okay, so that's so-called the splicing process. Okay, and then from that spliced mRNA, you can make a protein if it is used for translation process. So all these complex, identities of introns and multiple exons, you can only understand, if you understand the entire DNA sequence within your gene. Okay, so it's very important. So then DNA sequencing analysis itself, how does it work? There are multiple technologies or different techniques that can sequence out your DNA, and out of those, the very old but traditional DNA sequencing technique is called Fred Sanger's sequencing method. That's based on dideoxynucleotide. We'll talk about that first because that is a historical technique that's explained a lot in the biology textbooks and molecular biology as well, although it's a little outdated. but the principle is very important, because understanding that principle requires your full understanding of DNA replication. Just like PCR process, which requires your understanding of DNA replication, it also requires the same process. Okay, we'll talk about that, and then we'll move on to how that technology is modified. So we'll talk about the advancement of DNA sequencing technology. Okay. So how does it work? So if you use that dideoxynucleotide sequencing method, and back in the old time, about 30 years ago, you could about identify the sequence of about 800 base pair stretch through one analysis. Okay, for that process, you would have to do an in vitro DNA replication. So DNA replication happening inside of a test tube, just like PCR. Okay, and then once that's done, you will have to separate out replicated product on a gel electrophoresis to sort them out in their size differences. Okay, so that electrophoresis will be done on a polyacrylamide gel instead of agar gel, because polyacrylamide gel is more will provide you more resolution, higher resolution. So the replication, the in vitro replication to work, you have to provide everything that's needed for replication. So for example, of course, DNA template you have to provide, and a probe or primer. Okay, that's the single-strand DNA that can bind with the matching template DNA. Okay, primer should be given to the reaction tube, and then normal DNA nucleotide. Okay, GATC DNA nucleotide should be added to the tube, and then on top of that, you have to add a unique DNA nucleotide. So that unique DNA nucleotide is called dideoxynucleotide. So DDNTP, and means it can be one of the four. Okay, GATC, but dideoxynucleotide. So that's a unique nucleotide, which is different than DNA nucleotide. So we'll take a look at it. Okay, I'll show you the structure of the dideoxynucleotide. So this is a DNA nucleotide. Okay, so this is a deoxyribonucleotide because oxygen is removed here, so that's why it's deoxy. Deoxy means oxygen is removed, so one of the two oxygens gone. If you had OH here and OH there, that's ribonucleotide, which are the nucleotide used to build an RNA molecule. Okay, but if you want to build the DNA, that oxygen is gone, so this is deoxynucleotide, ribonucleotide. So these are regular DNA nucleotides that are used to build DNA molecules. But if you look at this, okay, the oxygen is removed from here and here as well. That's why it's called dideoxy. Two oxygens are removed. So this is a dideoxynucleotide. Okay, so you will add one dideoxynucleotide to the DNA replication tube. Okay, so once you do that, there's a very drastic change in your replication process. We'll talk about that. So you're going to provide that in a test tube that has everything that's needed for the in vitro DNA replication to take place. Okay, once that is done, and the replication will take place, and then whenever that dideoxynucleotide add is, actually your replication will be terminated. We'll talk about that process, why that's going to happen. And once that's done, and then all the replicated products will be separated out on an acrylamide gel electrophoresis. So they'll be sorted out based on the size, the smallest at the bottom, largest at the top, and in between they are separated based on the sizes. So you can sort out their sequences. That's how it's going to work. Okay. So the dideoxynucleotide sequencing method, that is the method developed by Fred Sanger. Okay. So that method utilizes DNA polymerases to synthesize new strands. So it's basically in vitro DNA replication process. Okay, new synthesis happens at the 3' end of a primer. That's the principle of DNA replication, right? You have to provide the primer, then the replication happens at the 3' end of a primer. Okay, and then DNA polymerases will add regular DNA nucleotides at the end of the primer. That's how it's going to elongate or extend the new strand. That's how it works. But if dideoxynucleotide is added during the replication process, okay, if that dideoxynucleotide is added, then that's when your DNA polymerase will have an end termination. The reason is, I'm going to show you why. So there's a nice animation that you can watch. So if you go to this website, so it's by copying, okay, copying this website address, okay, and then pasting it on a web browser, whatever you have, okay, paste that website address and enter, you can go to the website. So it has a nice animation that explains that Sanger's dideoxynucleotide sequencing process. Let's take a look at that. Okay, so this is a DNA strand. You can assume that this is a template DNA strand. So it's got a 5' end and 3' end. There's the orientation. Okay, so if you want to build a new strand out of the template strand, of course the DNA polymerase will do this work. This is the replication process, right? So it grabs the tail of the primer and then it fills by reading what your sequence you see from the template DNA. It's going to bring the complementary nucleotide and it's going to add it there. So if it sees T, it's going to bring in A nucleotide, put it there, and then if you see another T there, A nucleotide will be brought there, so there will be phosphodized bond made. That's going to continue. So that's the replication process. Okay, there's no problem with that. But if you look at that, so you know this is a phosphodized bond, okay, between these two primers. That's a covalent bond that's made. So in order for the covalent bond to be made, okay, you see the presence of oxygen there. Okay, you need the oxygen there. Okay, so the oxygen is brought by the regular nucleotide. Okay, let's take a look at the next slide. So again, same thing. If you look at the ribose sugar of the DNA molecule, you see the presence of oxygen there. Okay, and the phosphate, and then it goes to next to ribose sugar, the presence of oxygen there. Okay, so that is the case. So there is an oxygen there. So let's take a look at that again. So replication process. There you go, new nucleotide comes in, there's an oxygen there, OH. If there was OH there, yes, the phosphodiyester, the bond is made. Okay, let's go back and try it again. There's the OH there, right? Okay, and then the phosphodiyester bond is made, okay, between this nucleotide and that nucleotide. So the OH should be there for this polymerization to take place. Okay, so that is only possible if we have the regular deoxynucleotides in this newly made strand, right? So that's a kind of prerequisite. So right now, this nucleotide doesn't have oxygen there. So this is a dideoxynucleotide. Okay, so if you place the dideoxynucleotide, yes, it can go in there because it doesn't matter as far as it has a phosphate there. So dideoxynucleotide will be incorporated into a newly made strand. However, as soon as it's incorporated, since there is no oxygen there, when DNA polymerase will try to bring the next nucleotide based on what it reads on the template DNA, so there was a T, so it's going to bring in a nucleotide. But because of the fact that there is no oxygen there on this nucleotide, since it's a dideoxynucleotide, the phosphodiester bond cannot be made. Okay, so that's the result of it. So because of that, your replication is terminated, stopped right there. Okay, see the result? Okay, well, let's go back. So as soon as the dideoxynucleotide is incorporated, that's fine, but if you want to add the next nucleotide, there you go. It's impossible because there's no oxygen there. So replication stops right there. So what does that mean? I'm going back to the replication process. So in order for the DNA replication to work, you have to provide everything you need. What are everything you need? Template DNA, of course, normal DNA nucleotide, GATC, DNA polymerase, and the primer. Okay, if we have those, would it work? Okay, sure. So you can replicate, that condition 4 different places. You have a 4 tube with the same condition. Okay, and then you're going to take a look at it. Then each tube, you're going to add a dideoxynucleotide to each tube. Okay, GATC. Okay, so I'm going to stop this video right here. I'm going to start it with the next one.